the experiment was pioneered by Stanley Miller. Back then, who was simply PhD seeking graduate student at the University of Chicago at the time. But his academic advisor, PhD thesis advisor, was the renowned Harold Urey. So it's often called the Miller-Urey experiment. And then what he did was he devised the apparatus simulating the early ancient Earth's atmosphere, and then try to find out if he can collect some of the synthetic, spontaneously synthesized organic molecule, starting from simple inorganic molecule simulating the ingredients of the early Earth's atmosphere. So among many the such organic macromolecules, he was particularly interested in to see if he can find the synthesis of amino acid out of such simple inorganic molecules, because as we hopefully know by now, the amino acids are the building blocks of the proteins, and the importance of proteins are once you are able to synthesize spontaneously the individual amino acids, then you can expand your reasoning to the synthesis of proteins, and then some of those proteins should be able to work as enzymes. So that's the idea. So he built an apparatus simulating this, so this is must be a replica of his original apparatus, I guess. So in big ball of a flask, thing, he connected this electrode to simulate the lightning. Actually, later on, recently, it turned out that the lightning must have been extremely more frequent in those early ancient atmosphere, and then it should be able to provide ample enough energy source for this spontaneous synthesis. Was that something found out in later, very recently. So anyway, he had such an insight to simulate those lightning with this electrode to provide the energy of this synthesis, because since this has to be an anabolic synthesis, and what he had was simple ammonia and hydrogen gas and methane and some water vapors. Notice that there was no oxygen like the ancient atmosphere. It is a reducing environment. And then after a while, just after the cooling down this reaction, then at the bottom collect these samples, then wanted to find out whether in this sample if he could recover some organic macromolecule, in particular amino acid, any amino acid. And then wow, this is the chemical formula of the simplest form of the amino acid. So we don't really have to pay any other attention to this particular procedural things. After all, these are all the guessing procedure. It must have occurred in this type of a chemical reaction until it eventually reached to the synthesis of this, the simplest form of amino acid, which is actually happens to be a glycine. And then he was able to succeed this initially. And not only that, he continued on this experiment until he finally succeeded in synthesizing all these 20 standard amino acids. These are the ones that participate in the protein synthesis. So once you can demonstrate all this spontaneous synthesis of all these 20 different amino acids in the laboratory, that you know it speaks a big volume. You know, and not only that, some more complex form of carbohydrates also successfully synthesized later by himself as well as others who try to copy. Once somebody have a success story and everybody else is trying to copy that success, that's what happened in this occasion too. And originally his manuscript, his advisor Harold Urey was must have been really a fair, nice gentleman, not only a renowned scientist. He back then he must have already a Nobel laureate, if I remember correctly. Uh, so when he initially, after accomplishing his successfully his experiments, and then finished writing his manuscript to publish in any scientific journal, then he only showed his manuscript to his advisor Harold Urey, with his name also written on that together. And then Harold asked, "What's this?" And then Miller explained, and then Harold's response was, "Well, I didn't do anything on this, so I shouldn't be. My name shouldn't be here." So he just wiped out his name, and then tried to make it through to any respectable scientific journal. But then, unfortunately, all of those major renowned scientific journals declined his manuscript. So purely because who is this Stanley Miller? Nobody could recognize his name because he was a mere graduate student. So it couldn't even in the United States. That was 1950s. So those academic culture is always kind of a very liberal. Even then, that type of drastic liberal activity was not well taken by other established scientists. And then finally, he had enough. Then Urey decided to put his name also together on it, and then just like snap, immediately went through. And then this work came to known to the world, and then these two scientists became really famous, with is deservingly so. One minor point is actually, contrary to what most of the scientists have thought originally, actually the condition of this reducing in atmosphere was not really critical. So even with losing with the more loose conditions, like having some of the oxygen to allow some aspects of the oxidizing environment, that synthesis was still could be done, no problem. So that was it. So with almost always, the theoretical speculation and prediction does not match exactly with the real result. This also shows another such case. These Harold Urey has Getty's images in these days has everything. Oh, it's amazing. Why didn't I think of such a big business sector before? And Stanley Miller, although it looks so old, maybe it's just only because at the same time back then he didn't have any proper photograph of himself. Only after he became so famous now, trying to make up this trying to pose himself again and to make up the memory of this in front of a replica, it must have actually produced this photograph, I guess. So now afterwards, the more complex organic polymers and even those protocell or pre-cells were successfully synthesized in the laboratories. Okay, good, fine. However, the problem is, so this is a one such event of like protocell trying to absorb the RNA from outside into the inside to make this RNA. Then the RNA may be able to act from then on as their genetic material. Wow, impressive. And more impressively over here in this another type of the protocell type of structure generated in the laboratory, even was able to demonstrate very simple metabolic reactions. Yeah, it is to our standard, it is one of the such a simple metabolic process, but in itself, it is extremely sophisticated chemical reaction. And if these reactions occurred spontaneously in this structural setting, it is really something significant. So simple metabolism was even could be replicated in these things. But that was it. Only that was it. Well, but no one has ever been able to actually really demonstrate, hey, here is a real cell starting from such inorganic synthesis, organic, and there's or pre-cell formation, and then later this protocell evolving into more cell-like structure, and then now act as a living cell is probably what they must have really hoped to be able to show, but nothing. So unfortunately, no one could ever do that. So here, so the from the other angles, the other side of this whole thing, now this synthetic biology is becoming a very hot area in these days. So synthetic biologists are taking some lessons from the actual real biology, and they try to sometimes it's replicate totally synthetic approach using synthetic approach, or even try to create something non-existing thing, just simply taking some inspiration of this world, the nature showed us, is what basically the synthetic biology is trying to do. So one of the pioneer of this synthetic biology, he's not only the synthetic biology, but in many areas he has done a lot of marvelous job, including the human genome project. This approach taken by his name is Craig Venter. I casually compare him as the how can I forget his name? The late Steve Jobs. Oh my gosh. The Steve Jobs of this biological science field. He's an entrepreneur himself and runs fairly big this bio-related the company business. And one of his renowned story is related with the Human Genome Project, which here it is not such a good proper place, so I will not introduce his kind of involvement in the Human Genome Project. But other than that, he has done a lot of things, including this synthetic version. Now he was successful in creating version 3 of this synthetic thing, which means totally empty sac. What he did was a totally synthetic DNA, synthetic genome he created and then injected. Just like we are creating some empty vector and DNA vaccine and then try to inject into host to vaccinate, maybe a little bit of a similar approach. So then he was able to create this whole new organism. So this organism consisting of only less than 500 genes, to now it is the smallest genome organism in the whole world. We should recognize. So this way, try to also create new cells, but obviously this approach is totally different from those scientists who involved in trying to replicate the procedure of a chemical evolution, starting from the step they believe that it must have occurred in the beginning, right? So finally, I'd like to simply add on this RNA world saga to the first part of this lecture, this week's lecture. So as I briefly mentioned, originally the DNA was not really even the genetic material. On the contrary, what scientists believe is, instead of DNA, actually RNA was the first genetic material. Here, familiar, maybe we have seen this photograph of the RNA model, structural model of RNA in previous chapter, but a little bit different is now this RNA looks like what, a little bit chubby, right? RNA, previously the model what we have seen is a really kind of skinny, especially compared with the DNA double helical structure. But now this RNA even appears to be like chubby, and this one we have seen it already. Right? This RNA is being made here, out of this DNA template. This is a DNA template, and this is a specific enzyme. Guess what? What's the how would you name this? RNA polymerase, right? RNA making enzyme. So according to the complementary, this whole relationship is a complementary base pair between one strand of DNA and the now freshly being made RNA. Their relationship is complementary, their bases. So this RNA is being copied, so to speak, copied out of specific region of the DNA. We call it the DNA template, right? But getting back to this RNA world hypothesis, what it states is RNA was the first genetic material in the cell at first. Why does this type of speculation, maybe speculation or whatever, this theory was necessary? I mean, what's wrong with DNA? Because if DNA was originally the genetic material, you have a serious problem, as hopefully you can see in this figure. By the way, just right before I start this lecture, I recreated this whole the figure I drew myself. I'm so proud of this because the resolution of the original figure was so poor, so I didn't really like it. So I tried to copy and make myself the drawing of this using like basically a PowerPoint. I like it, so I'm happy. So Jesus, what a stupid. So here what you can recognize is at each step. So let's say starting from DNA, to continue your life form, you have to be able to do this DNA replication, and then with this extra copy you divide them, distribute equally each of them, so to partition into the two next daughter cell, the emerging from the cell division, right? But to do that, you need this DNA polymerase, the enzyme. Without enzyme, you cannot do this. You cannot even think about it, right? And also, to make this whole protein, necessary proteins, you have to start with this RNA making procedure called the transcription. Transcription is the procedure in which the DNA is being copied into the RNA, right? But to do so, you need RNA polymerase, the enzyme. And once you have this RNA, then using this RNA, you have to synthesize. Using the information of the RNA, you can synthesize only now protein. But to do so, you need this ribosome, which is basically whole mixture of protein. And it is an enzyme because this particular chemical bond, each chemical bond, the peptide bond, okay, which is a specific type of covalent bond or the dehydration reaction covalent bond, must occur in the ribosome. Okay. Now, you immediately realize that what you need is, because this DNA polymerase, RNA polymerase, and ribosome also takes proteins. But to do so, you need a protein, already pre-existing protein, but that's not, because you haven't even started your job yet. So there's no way you can make these proteins. So you have a big paradox. But then what would make what would RNA make any difference? Yeah, it will theoretically. RNA may be able to, idea is, may be able to function both as a kind of protein-like enzyme and RNA. Obviously, there is no problem for RNA to serve as genetic material, as you can see in these days, even in many different types of viruses, right? How can RNA be such a multifunctional, flexible in their function? Because first, this is an evidence of RNA being able to function some special, enzyme-like function. The ribozyme, the name was coined by this RNA plus enzyme, this ribozyme. So ribonucleic acid plus enzyme is just synthetic word of ribozyme. So it simply means that it is an RNA, but not a conventional RNA, but it acts as the protein, the catalytic RNA. So another name of ribozyme is catalytic RNA. So in 1980, something like was it 1986, around mid 1980, I guess. So anyway, these two scientists independently, one is at the University of Colorado at Boulder, and another at Yale University in the eastern coast. East Coast of the United States, independently discovered the presence of this ribozyme. So it turned out that actually ribozymes are something that is very exotic in nature. So only very rarely can you find in very some lower animals, very rare, some uncommon organisms. No, that is not the case, but these ribozymes can be found everywhere, even in these days. So it is not some kind of a fossil-like ancient relics, but in these days they are still functioning. Of course, functionality-wise, in most of the cases, protein may be able to function much more efficiently than as compared with the RNA. But the fact here, important fact that we need to recognize here is how come RNA can act as an enzyme. So once again, getting back to that, the popular, frequently used my point of emphasis that shape dictates the function is once again the theme here. So the reason why proteins, as I have already mentioned several times throughout this lecture course already, The reason why proteins can provide such a various function, including the enzymes and etc., is simply because of their ability to generate so many complex, diversified, and specific function, shape. I mean, not function, the shape. Okay, but then RNA can mimic this. Unlike DNA, which is boring, but RNA can have this kind of folding, three-dimensional structural shape folding. We call that as a secondary and tertiary structure. So here, if the RNA was generated as a result of transcription, you can see this is you can recognize this as an RNA because once you see this this U, so it must have been RNA, right? And like I mentioned earlier, RNA for some reason tends to remain itself. RNA doesn't like to form, unlike DNA, RNA doesn't like to form this twisted form of a double helical, the complementary strand. Rather, RNA just wants to keep into himself or herself, should I say? Well, itself is more appropriate. Yes, itself. By making this so-called intra-strand complementary base pairing. So basically they are establishing the complementary base pairing between the specific base pairs like G and C and A and T internally within some of the regions, like as you can see. So this region, where are you? This C, and this probably this region and that region somewhere over it, and then they can have this specific interaction, and those who cannot have such internal complementary structure will loop out. Then simply by looking at this 2D structure, you immediately see there is some kind of a shape is generated. But then we also know that this will not only stay in this like two-dimensional area, but actually this process will involve the entire three-dimensional structure. So now you will have another tertiary. So this is called the secondary structure. This is a secondary structure, but then if you view this from a 3D angle, then it will become the tertiary structure like this. So this is an example, structural model of the famous transfer RNA, one of the RNA, one of the many different kinds of RNA in our cell. And its function is, as we have seen earlier in the previous chapter, transfer RNA's function is to carry individual amino acid to the site of the protein synthesis, which is the ribosome. Okay. So it is an extremely important component in our cell for our survival, and it has specific shape, starting from this type of a specific secondary structure formation through this intra-strand complementary base pairing. Internal complementary base pairing is instead of extra the strand, and so it generates specific shape. Then it can have an opportunity to brag, show off any unique function. So that's exactly what's happening. So it is no better and no worse. Exactly what it looks like. And so this is one famous example of why enzyme and how enzyme works. And we have similar movies even in the previous the chapter lecture. So some specific shape recognition and fitting in, and within that you can perform any kind of a meaningful function. And many such RNAs being able to generate such unique shape using their own unique such folding structure, so then they function as ribozymes. So many different types of ribozymes discovered, and you have some kind of decent repertoire of them. Some of the structures with their names are shown over here. Obviously, you don't really have to worry anything about their names and functions or whatever. It just there are many different ribozymes out there. So that's the proof of RNA there must have been able to function as a, because if that was the case, then RNA at the same, at least temporary basis, they could because they are able to form this complementary base pairing, they can replicate themselves, just like modern days RNA, the RNA viruses do. And also at the same time, they can do this perform their own catalytic function, and until more stable and sophisticated proteins could be generated by such an action. Of course, it must have taken such a long time until this actual transition was made from RNA to eventually DNA, and then from DNA now RNA to proteins type of the way it works as we know in these days. So RNA is great things. It is a multifunctional things. But then why? The question you might want to have this type of question. Then why did we switch? RNA seems such a wonderful amenable ability. So why did we dump them? Well, there was one serious problem. Chemical stability is among many other things. RNA has some other deadly weaknesses, but this is actually the most serious problem if we decided to stick with RNA as our genetic material, because RNA is chemically very unstable, so it is unreliable if we consider RNA and then simply put our whole eggs on RNA, then RNA would have easily broken. Yeah, literally, it's very easy to break because of this presence of this extra oxygen, where DNA in the ribose sugar, this oxygen is not there, right? But in RNA, oxygen is there, and this oxygen, wherever, anytime you have an oxygen, you have to approach with caution, because you never know when this oxygen is going to make troubles. This oxygen will attack this. This oxygen will attack this. So, resulting in cutting of all this covalent bond that enabled this assembly of nucleic acid out of nucleotide. Then we have a problem. DNA doesn't have such problem because of this lack of this oxygen there. So it is naturally, it must have something that happened that gradually, naturally, quickly, you just go away, not here. DNA must have been able to replace the position of the genetic material. So in these days, actually, RNA function as a regulator of protein synthesis. So here it shows three such types of RNA related in the functions, whose functions are related in the protein synthesis. As messenger RNA is the carrier of that protein information, because it is a direct copy of the gene. Ribosomal RNA, on the other hand, is an important component of the ribosome. So without the ribosome, you cannot do this any protein synthesis. And transfer RNA, as I have said just a minute ago, it is the RNA that carrying individual amino acid to the ribosome. So all these three are essentially absolutely required for protein synthesis. Although this is the only one actually directly bear the information of the protein actually, but these two are nonetheless also equally essential for that procedure. And also another thing is that these three are not the only types of RNA we have in our cell. There are three more different types of RNA that I didn't bother to bring them on to introduce to you because this is not the proper time. So this is the classical three version of RNA. But then later on, when I was in my days of about the graduate school, those three new identities of RNA became known to the scientific world. So later we will have an opportunity to at least to hear their names about. And here this structure is the model structure of a ribosome, inside of which many protein and several specific type of ribosomal RNA is there. The point here is ribosome itself is actually ribozyme, some specific RNA in the ribosome is the one actually make this catalysis of connecting individual amino acid. Surprise! So even the ribosome is actually ribozyme is the message. So probably I wouldn't really stay over here to let you digest this silly cartoon about this ribosome. Ribosome is complex is the message right over here. And I really like this cartoon of the ribosome structure. Later in chapter 5, I guess, we will be able to see this shape with their related function once again. Actually, in the next chapter also, there is an occasion that we also get to see the structure of ribosome, but not related in its respective function. So, continuing on to the next section, which is about the core of this biological evolution. So we are going to take a look at the the steps by which all these diversified species arose. Although briefly, we cannot really pay too much details on it, and it will take forever. So just like at least try to scan the surface scratch, so to speak. So this photograph illustrates such a small aspect of diversity everywhere. So we can easily notice that so many different animals in the form of the variety of animals and plants. So in this small local area, can you find so many different sort of species of different organisms? Not to mention that if you consider in the water, inside of the soil, how many different types of numerous microorganisms, including bacteria and protists and so many things. And once again, the butterflies and mosses are also very small group of the small part of this huge animal lineage, but still you can easily notice that the diversity, you cannot miss the diversity in anywhere, everywhere. And perhaps we can actually emphasize throughout this the study of this chapter, we can also remind ourselves again the importance of this species diversity and what happens if we lose the grip of maintaining the species diversity. It will all come back to us to backfire and endanger our own well-being. So the first living organism like Luca type of things, the first several generation, or maybe more than that, all those organisms at the time still the single-celled organisms like a prokaryote. Yes, prokaryotes are fine. We can understand. The first form of life, the cells, were prokaryote, not having the nucleus and the cellular internal organelles yet. Okay? But then, what is this? Heterotrophic prokaryotes. So there are two different kinds of, according to their mode of nutrition in metabolic spectrum, there are two different types of organisms. The one who can make their own food and the one who cannot. And these heterotrophs are those who cannot make their own food. Okay? So it is a kind of a more straightforward, easier, common sense version of the definition of heterotrophs by saying organism that has to obtain its food from other organic materials. Well, sounds like kind of a just the type of so obvious things. However, if we think a little more details about it, these organic materials, food, in general, foods are all organic material to everybody else. Okay? Why? Because organic materials are the only ones that have enough energy inside of their chemical bond. So the only purpose of a food is so that once we have those food, then we break to release their energy, store the energy so that we can convert those energy into some other forms. So still, we are perfectly obeying the first law of thermodynamics here. Okay, so that's the purpose of the food. But then what do you mean by that food? Those who can only have their food in the form of organic material. Are there any others that who don't have to? Yes, there are. So we'll get to that later. So heterotrophic bacteria and protists, and also animals and fungi are those who belong to the heterotrophs. So these are like the reason why they can, they have to get their food in the form of organic material is simply because they cannot synthesize the organic food on their own, starting from inorganic material. But the other types of the organism, which we call as the autotroph, can. So inorganic material, if you can convert this inorganic material into organic, then it will become food. Yeah, it is food. To do so, you have to provide what? Energy. Right, of course, because from inorganic into organic, it is an anabolic procedure. But still, even with the, yeah, of course you can, once you have this inside, when you have plenty of things, some of you may be able to do this, but to say perfectly accurately, those heterotrophs cannot do this starting from inorganic material into this the form of organic, so that you can pick the carbons, then accumulate those carbons into the organic molecule must contain plenty of carbons, right? But then think of then if you can start this procedure starting from inorganic carbon, which is carbon dioxide and stuff like that, and then it will be a tremendous plus. But unfortunately, these organisms labeled as heterotrophs cannot. So that is their very sad fate, because for food they must obtain in the form of organic materials, but then the only place they can find any organic material is in the body of other living organisms or those who once lived. So in other words, we have to, as heterotrophs, we have to, inevitably we have to sacrifice others' precious lives to sustain ourselves. Personally, I don't like that, but that's our fate. So anyway, so for even prokaryotes, here what the message delivers is, even for prokaryotes, there are some prokaryotes are heterotrophs, and protists, same thing. And protists and animals and fungi are all eukaryotes. So, like, there is only one very notable exception here, which is missing in here, which are plants. Plants are like a very distinct representative of the other group, the autotrophs. So let's take care of this heterotroph first. So even for heterotrophs, the mode of nutrition for animals is somewhat different from that of fungi, although both animals and fungi are multicellular, unlike the bacteria and the most of those protists. These are multicellular organisms, but as heterotrophs, the way they obtain the food is, in the case of an animal, they obtain the food first. In other words, ingest, which means internalize the food inside, and then they do have this special organ for the digestion, like a stomach or intestine, and then they digest this food internally. Okay, that's the way they handle their food. On the other hand, in the case of these heterotrophic fungi, all fungi are heterotrophs, right, including the yeast. They digest the food first externally and then absorb. In other words, even if they do not take all their food inside, some of them are already like converted into broken down form outside. So it really helps to decompose so that they can recycle. They can help recycling those material in Earth. So they are one of those so-called decomposers. They help recycling of this whole material in the world because of these differences. Now, in the case of the autotrophs, those organisms that can synthesize their own food from inorganic material, which is great. Wow. So although we think one of the like whenever we try to think of the autotroph, we get to remind ourselves of this green plants and photosynthetic algae, like those who make our beloved the rivers like full of this green latte. Okay, but anyway, this photosynthetic algae and plants are the two major representatives of autotrophs, those who are capable of a specific mode of this autotrophic metabolism. But the point is, the photosynthesis is not the only way of doing this autotrophic metabolism. There are some other bacteria, although some bacteria can do this photosynthesis, but some other types of bacteria do the similar type of autotrophic metabolism, but is not dependent upon the sunlight or any light energy, and their mode is somewhat different, although it is a little bit more primitive form of this autotrophic metabolism. They are called chemosynthetic bacteria. So in photosynthetic, if we describe the reaction, it can be summarized like you take this carbon dioxide, usually from outside in the air, and you take some of the water, and voila, you build the big, relatively speaking, complex carbohydrate like this is glucose, one of the monosaccharide, one of the simplest form of the sugar, but still starting from carbon dioxide, which is the inorganic molecule, then you convert them into organic glucose, is something really, really remarkable. But additionally, you have this byproduct. Oxygen gas is released, evolved, okay? Evolving the oxygen gas. Here, the evolving means that doesn't mean that evolution of the biology, evolving the oxygen gas into the atmosphere, so that that's the reason why in these present days Earth is filled with so many oxygens. Should I say so much oxygen? Maybe. But I will consider the oxygen as an individual molecule, so many oxygen is still valid grammatically. And because of that, actually, it has a tremendous influence on the lives of the other form of lives afterwards. On the other hand, the chemosynthetic, the autotrophic metabolism does not release the oxygen gas, but it's releases a similar compound of the sulfur. So to us, doesn't help at all. But still, very similar metabolism, and they what they want, they get what they want in this case. So here, although I show the two different type of chemical formula, but for your own sake, you don't really have to really concern about the details of this chemical formula. But the one thing is, during the photosynthesis, which is a type of autotrophic metabolic reaction, the oxygen gas is released. Oxygen is produced as a byproduct. It is a kind of a trash for them. But to us, the special form of these heterotrophs, the oxygen is a life to us. This kind of a really interdependent way of living has been established afterwards. So in like a continuation of the original the line of this statement. So once all those life form at first was heterotrophs, but then some small subgroup of those heterotrophs, out of them, new prokaryotes, new bacteria who are capable of this, the photosynthesis, arose. How do you know? Many different evidences actually you can get, so that there was some photosynthetic prokaryotes evolved sometime afterwards of the original first type of prokaryotic cells on Earth. But then there are the present days descendants of this original photosynthetic prokaryotes. Their names are cyanobacteria. This cyanobacteria hold on for a minute. Yeah, there we go. This cyanobacteria in Korean, because someone actually have asked, so I'm trying as much as possible. I'm trying to give you some of the terms in Korean version. It's called, if you can forgive me for having such a poor handwriting, I will just gladly providing you with this some of those terms, Korean terms, for each English terminologies. So cyanobacteria are the first type of, probably those ancestral form of these cyanobacteria must have been the first type of those prokaryotes who were capable of this photosynthesis and released the oxygen into the atmosphere, So as a consequence, now gradually, slowly but surely, oxygen gas became accumulated in the air. So these are two different types of such photosynthetic bacteria. Although these cyanobacteria look green because the light capturing pigments are very similar to those of plants, green plants, but that does not mean that these purplish looking bacteria does not produce any oxygen. They do also, although the difference is they possess some little slightly different types of light capturing pigments in different colors. That's all. But by far the majority of this speaking of this bacteria, the majority of photosynthetic bacteria are the cyanobacteria. And another things of these cyanobacteria are they appear to be this multicellular form. Here, something like, if you look at, what is this? You have so many different cells can you see in each this is one cell, 2, 3, 4, 5, like this. So one may say, hey, isn't this in the multicellular form of life? You said, apparently the other day, you said no bacteria are multicellular, only eukaryote, only some of the eukaryote can be a multicellular. So you lied once again. No, it is not. I told you the truth. Even though they are multicellular, look like multicellular in form, what did I say about this multicellularity? Without any differentiation. Without differentiation, no, you are simply nothing but, you are not multicellular, but simply a cancer. I'm not saying this cyanobacteria is a cancer, but speaking of this multicellular, like eukaryotic organism, just because you have lots of cells clumped together doesn't mean a thing. Is actually, if you look closely inside internally, these cyanobacteria, they do have this all this green area, membrane-like green area where this photosynthesis is taking place, actually. So these days, present days, you can see a lot of these cyanobacteria form of some of the layer-like, fossil-like, very similar to fossil, the dead layers of cyanobacteria, and on top of that, the new layers of the cyanobacteria like begin to prosper type of structures can be found on many different areas of the beach, and they are called stromatolites. But then the point is, similar to these stromatolite layers, in the early long time ago in the early Earth, and some of these fossil forms, people were able to discover very similar cells, which is very much look like this present days of cyanobacteria stromatolites. So that's, you can actually kind of figure that, oh, those must be the ancestral form of this present days cyanobacteria. So we can have a little bit of a very simple summary of timeline through which what happened in the form of in the case of this evolutionary transition. So according to that, like about 4.6 billion years ago, by the way, this BYA stands for billion years ago. So it was about 4.6 billion years ago. It was like it's been estimated as the age of Earth, and then about 1 billion years later, then only can we find the first form of life in the form of these prokaryotes, like this. wow, it's 10 billion, I mean 1 billion years. Uh, can you imagine how long that 1 billion years period would that be? It's just unimaginable what it's like. But then another 1 billion years has passed until can we see the first form of the first eukaryotic organisms, even much more than 1 billion years. Actually, this is after about 1 billion years later of the first emergence of the prokaryotes, which are, like I said, if you take my words, which are all the heterotrophs. But then the first form of eukaryotes, the evidence, how do you know? Oxygen. Now the evidence of oxygen accumulation for the first time, and the Earth's record. Okay, can you find? So it took such a long time for this new type of the organism capable of autotrophic metabolism, and as a consequence of their diligent efforts, now Earth became the oxygen rich to a certain degree. Probably at that time not as much as the present time, but at least there was a start. And then still after a long period, then now first eukaryotes. Okay. What's the point of this all eukaryotes in these days? What's the point? The characteristic feature that every eukaryote has is the presence of what? Mitochondria. Yes, the mitochondria. All the eukaryotes have this specific organelle, the mitochondria. Its function is to generate a huge amount of the ATP, right? ATP. Hopefully you know what the ATPs are. These are the convenient form of the energy. And they actually, presence of the mitochondria revolutionized the metabolism of this energy synthesis. So even without the mitochondria, still cells could produce this ATP, but that would be the presence of the mitochondria just changed the way they can. The efficiency of production of ATP has been simply just a night and day, or heaven and earth, like the difference. So they could be in size, they could become bigger in size, they could afford the bigger, like if only when you earn enough amount of money can you move into the bigger house, like a bigger apartment, like so to speak. So then you have to fill in all those internal huge area of internal inside of your apartment with so many different kind of furniture, just like the eukaryotic organism does in the form of organelles, maybe. So then still some long period of time later, now first form of the multicellular organism have emerged. So the big point is why this multicellular organism do we have to so much wait so long time until we get to see the first appearance of this multicellular eukaryote. The first is why only eukaryote can afford this multicellularity. The second question is why only so late this multicellular organism, eukaryote could be possible. I think it is all about the oxygen, because once you have accumulated huge amount of oxygen in the atmosphere, so then all these organisms could afford the upgraded, much geared up, higher activity of this metabolism utilizing this mitochondria. So then only then they could afford this huge amount, much larger amount of the energy spending is the reason. Another thing actually the oxygen has contributed so much is like they allowed the transition of the oldest organism from sea to land. Before the oxygen has accumulated enough, still there might have been some brave attempts of different organisms to make their move onto the land for many different reasons. So, for example, like on some of the area, like near the seashore area, might have been some drought, so they had no choice but to simply move somewhere else, and then their obvious choice, reasonable choice, must have been some inland. But before those accumulation of enough oxygen, all those efforts might have been failure. None of them would have been able to safely settle in on the land, some area of the land. Why? Because of this the ozone layer, the enough buildup of the oxygen gas up in the atmosphere, in the stratosphere especially, then they form some protective layers of ozones through this spontaneous chemical conversion. And this ozone layer is the one actually, we all know that they can protect all those hazardous lights, including X-rays and UV light and many different things. So without ozone layer, even in current present days, our lives would have been in great danger. So that was another very significant contribution of this oxygen accumulation on on the earth. So that's one thing, and another thing is, as I briefly mentioned once again, is due to this availability of a larger amount of oxygen, with the mitochondria being able to produce more amount of the ATPs, now it's possible for them to become larger, okay? And not to mention this multicellularity. One such like a showcase can you find in the early evidence in the form of these fossil records. Here, when we plot this timeline on the x-axis and on the y, when we compared the relative oxygen level in the atmosphere along this different timeline, and here this is the approximate level of the oxygen present days. So after the evolution of the first photosynthetic bacteria, now this oxygen level began to spike gradually, and actually something that I also probably should mention is probably somewhere along the line the big chaos must have occurred. So the pre-existing many different types of organisms, those who are not familiar with the oxygen, so there must have been great mass scale extinction must have occurred. The extinction, great, probably in terms of the numbers, the greatest extinction might have actually occurred over here. Due to the oxygen. So oxygen killed those who cannot adjust their lives into the oxygen. A lot of those existing animals must have actually, and not animals, organisms must have hated the oxygen. only those who can utilize, take advantage of the oxygen could survive. That actually has been also recorded in the form of all those different fossils. But another interesting record here there were some period, although it was relatively brief in the geological time scale, the oxygen level was something that much higher than the current level. We do not know why that such a higher oxygen level suddenly dropped like this, but very interesting coincidence was at the top, at the pinnacle of this higher level of atmospheric oxygen, there were such a huge gigantic flying insect. Like the gigantic flying insect, what we are talking about is this big size of the dragonfly, like wow. So the reason why they were available at that period and then all of a sudden they got extinct as the oxygen level dropped like that is probably the only plausible explanation. I shouldn't say the only, but probably the most plausible explanation is that the high oxygen level enabled them to have such a much higher energetic, like metabolic activity, especially if you consider that those flying insects, they need much more amount of this energy expenditure, even much more than the birds and any other really actively moving animal parts. So only those period, such a big-bodied flying insects were capable of maintaining their big-sized body and then their energy demand. But then those supply of this energy in the form of the oxygen dropped, then they cannot afford to sustain themselves. And that's the reason why they all had to go, is probably. And speaking of the extinction, well, there was the most recent the event of such extinction, although this is not the only event. We all know, hopefully, that what happened here. Some meteor hit specific area near the Mexico, Yucatan Peninsula, this particular area of Chicxulub crater. Only later from satellite can the evidence of the crater be found. Only in 21st century they were able to precisely recognize and identify the details of. Before there was only rumors going around, but could not really recognize and confirm precisely. So this is called something like a KT extinction. So if we actually click on, where it gone? Yeah, there was what is some advertisement. I hate advertisement. So this KT extinction in the Britannica online can follow. Well, more recently it's called the KP extinction in a compliance of this new way of recognizing and naming this different geological timing. So nowadays it's called, actually it should be called the Cretaceous Paleogene extinction instead of a KT, previous version of a KT extinction. But either way, anyway, what happened was at the time this big impact generated such a huge disaster, not only in this nearby area, but the entire the global scale for the following many years, involving huge disasters, chaotic scale of this climate change. And so the majority of organisms could not survive, including the majority dominant ones at the time, the dinosaurs, are all wiped out except for the very small number of small the branches of such dinosaurs, including like the crocodile and etc. Right. Uh, but birds were able to survive somehow, and so the evidence like the rare metal iridium, which is very rare on Earth, but at the very specific period of the geological time, at the boundary of this KT boundary, these layers of iridium was found worldwide. In other words, the dust, impact dust of this meteor, spread it all over the world and then settled down, and then the geological sediment covered up afterwards. That's how you have these distinct iridium layers like this. And another evidence is this so-called the shocked quartz, one of the the rocks only happened after the extreme high pressure created by just like this huge impact, like such as a meteor hit, were also found the worldwide. Wow! So in other words, you can actually imagine how big of the impact was. So no wonder why almost most of those organisms living at the time just wiped out. Another thing is like locally around this impact area, this Gulf of Mexico, there was a really specific glass. It's like a remnant of this glass-like small. This was the rocks, the molten rocks, due to the high pressure and the heat of the impact. Molten rocks rains and then settled on the ground. It can still be found in near the area of those impacts. So this is one of the very famous particular type of organism, animal, ammonites, has been all wiped out, extinct due to this impact. Before it was one of the predominant marine animals, but it's all gone these days. No, not a single surviving one could be found. And not only that, so here there's a record of so many different impacts. The size of the impacts are represented by the size of the circle on this map, and so this is that particular impact created that KT boundary extension. But what we can see on this map is that was not the only impact. Look at so many different impacts of this one was like a comparable size and small scale size of the impact. The point is the Earth has been hit numerous times from the material, whatever, this meteor or asteroid. So many different types of impacts been recorded on the Earth. In other words, if that was the case, then another one will surely come. It's just a matter of time. And then we may also have to go through the same fate as all those poor dinosaurs had to experience sometime in a future, whether it's a near future or a distant future, or just we have no control over it. So here are the records of so many different the extinction. Like, so here what we can see is this was not even the largest scale of this mass extinction, and before there were so many other greater scale extinction has also been there. So with that, the only reason why I am just finishing this lecture with this gloomy extinction history is because later, not today, but later, continuation of this chapter, the next topic is highly related with this extinction. Once you have this mass extinction, then although it is really unfortunate for those pre-existing so-called dominant living ones, but it's now all of a sudden it's just entirely opening new opportunities for those once minorities. Now it has a tremendous opportunities to become expanded and then to become diversified. That is something that we are going to follow in the next lecture. So for now, I will just have to unfortunately say goodbye. But I really have a strong urge to continue some of it and finish some more materials to include within this week's lecture. But I actually will refrain myself not to, because some of the students actually kindly advised me against it in the previous week's our recitation Zoom session. So bye bye.
In the 1970s, a scientist named Jack Sookmyung embarked on the most ambitious fossil hunt ever. But instead of digging in the dirt, he dug through dusty piles of academic research and scraped together a record of all the ocean-dwelling creatures known to science. It was tedious work, but the data it yielded gave rise to perhaps the most famous chart in paleontology, a tally of marine life over time that revealed 5 catastrophic global biodiversity crashes in Earth's past. Those crashes, known as the Big Five mass extinctions, are commonly believed to have been the biggest die-offs in Earth's history, both in and out of the oceans. They include that time 66 million years ago when a huge meteorite took out the non-avian dinosaurs, as well as the even more apocalyptic extinction at the end of the Permian period, which wiped out more than 90% of Earth's species. But the Big Five weren't the only big mass extinctions, and they might not have even been the biggest. Take the Great Oxygenation Event of 2.3 billion years ago. Evidence from ancient rocks tells us that levels of atmospheric oxygen spiked around this time, poisoning the oxygen-hating microbes that had dominated Earth for well over 1 billion years. And yet, we can't really say how that extinction compares to the Big Five, because its victims were microscopic and left virtually no signature in the fossil record. In fact, the fossil record only captures a tiny fraction of the life that has ever existed on our planet, but it also captures a different fraction from one period to the next. In general, the record gets less and less complete the further back in time you go, but that trend is complicated by several other factors. For one, we have a lot more fossils from swampy places and periods of the past, not necessarily because those periods were more diverse, but because the calm, muddy conditions were perfect for preservation. We also have a lot more rock from some periods of time than others, and in general, the more rock we have, the more fossils we find, and the more abundant life appears to have been. In other words, the fossil record is inconsistent, so we can't just tally the number of species and take the results at face value, as Sookmyung's Big Five chart did. But paleontologists have used some clever statistics to adjust for those inconsistencies, and the latest diversity curve makes it look like there have been 8 major mass extinctions rather than 5. But you could also count 11, or just narrow the list to the 3 most massive. In short, it's pretty arbitrary. What isn't arbitrary is that some mass extinctions alter the evolutionary tree of life far more radically than others. For example, the first of the Big Five pruned away more than 80% of the species on Earth, but it left all the big branches of the tree intact, so life went on more or less as it had before. Other mass extinctions, even some smaller ones, like the one 66 million years ago, trimmed the evolutionary tree far less evenly, decimating some previously thriving branches and preparing others for world domination.